Have you ever wondered how reliable self-reported coffee consumption is in health studies? It's a fascinating question. Picture this: millions of people around the globe savoring their daily cup or cups of coffee. This popular beverage, brimming with bioactive substances, often finds itself at the center of scientific studies exploring its effects on human metabolism. But here's the catch. The data on coffee consumption in these studies often depends on self-reporting by participants. That's right, we're relying on individuals to accurately recall and report their coffee intake. And as you might guess, this method isn't always spot on. This discrepancy can potentially impact the validity of nutritional studies, raising concerns about the credibility of the findings. So the problem begs the question: if self-reporting isn't reliable, what can we use to verify coffee consumption? Imagine if we could use biological samples to distinguish coffee drinkers from non-coffee drinkers. That's where biomarkers come in. Biomarkers are substances that can be measured in the body and act like a biological fingerprint, revealing patterns of behavior such as coffee consumption. However, identifying a reliable biomarker for coffee isn't as simple as it sounds. Currently, only a handful of substances are known that could potentially serve as coffee markers. But there's a catch. These substances are yet to be fully validated and aren't available in sufficient quantities to serve as reference substances for comparative measurements in nutritional studies. This is a hurdle that needs to be overcome before these substances can be used effectively. One potential solution lies in the roasted coffee compound N-methylpyridinium, a promising biomarker candidate. So, what makes N-methylpyridinium a suitable biomarker for coffee consumption? Well, let's delve into the science behind it. N-methylpyridinium is a specific compound found in roasted Arabica and Robusta coffee. This isn't a random choice, mind you. It was first proposed as a biomarker candidate by researchers at the Technical University of Munich back in 2011. Fast forward to now, a team led by the Leibniz Institute for Food Systems Biology has painstakingly validated this unique compound's suitability as a biomarker. Remember, a biomarker is a measurable indicator of some biological state or condition. In this case, we're talking about coffee consumption. The team's validation process was comprehensive, involving an analysis of existing literature data and biological samples from over 460 individuals. These participants, from Freising and Nuremberg, had previously taken part in a nutrition study conducted by the BMBF-funded Enable cluster. Now, here's what makes N-methylpyridinium stand out. Firstly, it's chemically very stable. This stability means it isn't easily altered or degraded, making it a reliable marker. It's also not just randomly absorbed into our bodies. Its absorption is concentration dependent, meaning the more coffee you drink, the more of it gets absorbed. But how do we know if it's there? Well, N-methylpyridinium can be easily and reproducibly detected in various body fluids after coffee consumption, and it doesn't hang around for long. Within a few hours to days, it exits the body unchanged in the urine. So even though we can't determine the exact amount of coffee consumed, N-methylpyridinium allows us to distinguish between coffee drinkers and non-drinkers. We don't have to rely on memory or honesty. Instead, we have a scientifically sound, reliable method to tell who's been sipping on that morning brew. This makes it a reliable, qualitative biomarker for coffee consumption.